Yes, hi everybody. My name is Sarah Kuschert, and I'm currently a fourth year PhD student at the University of Queensland. I work at the Centre for Advanced Imaging, and my PhD supervisor is Maddie Mobley. So I'm very excited to be able to talk to you today, and feeling quite lucky given the current frosty relations between Australia and the British Isles. So very much feeling excited to present today. I'd like to start off by having an acknowledgement of country. The University of Queensland acknowledges the traditional owners and their custodianship on the lands on which the university stands, and we pay our respects to their ancestors and descendants who continue cultural and spiritual connections to country. We at the university recognise their valuable contributions to the Australian and global society. Now, non-canonical amino acids are often found in shorter peptides that act as natural bioactive substrates of various biological processes. So NCAAs are extremely prevalent in various medicines. For example, a lot of antibiotics and immunosuppressants, a lot of, as I said, natural products. So there are various different ways that peptides containing non-canonical amino acids are produced. And they are through non-ribosomal peptide synthesis pathways, or through standard peptides or proteins that undergo post-translational modifications, and as well as artificial chemical synthesis. So, as I said, these NCAAs present a large pool of biologically active molecules. And these include some examples that I'm going to talk about today, such as tyrosine, vancomycin, cyclosporin, as I said, antibiotics, and immunosuppressants. So when we come to studying peptides with NMR, however, we do see that there are some limitations when we're wanting to study non-canonical amino acid containing peptides. So NMR, as we're all familiar with, is an incredibly useful tool when determining the 3D structure of proteins or peptides. And many different software exists for transforming NMR data into a structure. So some of you may be familiar with CNS, ARIA and Explore. Some of you may be familiar with Cyana. In our lab group, we work primarily with Cyana. And the process for structural determination comprises of mainly two distinct steps. That is, firstly, the collection of structural restraints through experimentation, and secondly, using those restraints to calculate a structure. So in terms of that first step, peptides that contain non-canonical amino acids are really well suited to NMR structural characterization. They're small in size, and their side chain to side chain links that are often present induce local structure, which is fantastic for NMR to be able to detect. However, there is an issue when it comes to that second slide, using a software to be able to decode or implement these restraints to find a structure. Of almost 200,000 structures in the protein data bank, only 11,000 were annotated with a post-translational modification. One of the reasons for this is NMR relies heavily on modeling software to transform experimental data into structural models. And when these modeling software do not have access to create non-standard amino acid templates, then you're left with a case where you can't then incorporate those into your structure calculations. So I'm going to frame this mainly around Cyana. As I said, I'm fourth year PhD student, so I've been working with Cyana for about 4 years. Unlike the other software that I mentioned, Explore and ARIA, Cyana operates in torsion angle dynamic space. Basically, bond lengths, angles, chiralities, and planarities are kept fixed in optimal values. And this is often selected because it's a lot less computationally expensive. I think degrees of freedom are vastly decreased when you keep these fixed. But because of that, it relies on a set of really stringently defined library files, one for each amino acid and a couple for some commonly non-standard amino acids. However, at current standing, there are 27 amino acid library files in Cyana, which falls well short of the plethora of non-standard amino acids that exist in nature, and that's before you even get to some of the things that chemists and others can dream up and try and put into their chemical synthesis of novel amino acids. So Cyana requires an amino acid sequence as well as NMR data to be able to create a structure. And as I said, there are some current limitations of Cyana. So when it was first released, it did not have a method to create library files for non-standard amino acids. Additionally, even as it stands, creating NNC terminal specific library files is extraordinarily difficult. There isn't an ability to introduce a backbone modification to standard amino acid library files, and there also isn't a method for creating complex side chain to side chain bonds. So when we think back to how many biologically active peptides there are that do contain non-standard amino acids, not being able to have these, having these as limitations severely cuts off the use for Cyana in delving into those various forays of structural determination. So in order to address this, Peter Kuschert, who is the author of Cyana, in 2015 released an algorithm called scilib. It is able to create a non-standard amino acid library file if a chemical component directory is available for it. It's a fully automated process, not simply a format conversion, but it is a little bit of a, if that file is available, you run it through scilib, it produces a library file, which is then amended to the Cyana library. But if a library file doesn't, if a CCD file does not exist, it can't be run through that algorithm. And that is where our work began. So we're fortunate enough to partner with the Molecular Dynamics group here at the University of Queensland, run by Alan Ma. And we used their automated topology builder, which is publicly available web server and allows users to generate optimized geometries and topology files for popular MD simulation software. So these topologies are generated using various levels of quantum mechanical and semi-empirical calculations. And despite that being very out of what I'm familiar with, when we sat down with them, we did realize that these topologies and geometries are suitable as template files for NMR structural calculation software. In fact, they were already producing those CCD files that you need to run through scilib. So with Peter Guntert's permission, we integrated scilib into the ATB and created a system through which you can design your own non-standard amino acid, upload it to the ATB, and then have that provide to you a Cyana library file. However, there's a little bit more to it, and that's what I'll be outlining for you today. So as an ATB user, it is free for academic use. What you can do is provide a set of 3D coordinates. We often recommend using a PDB file, one that you can build, say, in PyMol, a set of atomic connectivity data, which is provided in that PDB file, as well as providing the overall formal charge of the molecule. So an overview of our pipeline is that we can build a non-standard amino acid in a tripeptide template, upload it to the ATB, which undergoes several steps of processing, and then provides to us the 3D coordinate PDB file, which can be used in CCPNMR, as well as Cyana files, which can be used if your software of choice is Cyana, and parameter and topology files, which can be used if your software of choice is of the CNS variety. There are additional things we have undertaken to extend the use of these programs, such as creating a process for creating sidechain to sidechain links, as well as creating a naming algorithm which assigns appropriate atomic names to amino acid side chains. So firstly, in that upload, you need to provide your NCAA in a tripeptide form. So it's produced with I hope you can see my mouse, an ACE cap, an alanine cap. Then you build your amino acid however you want in the middle, another alanine, and then an N-methyl group to finish. With these, this is then saved, when I build it in Kymol, I save it as a PDB and upload it to the ATB. You can also build terminal amino acids to be recognized using an N or C terminal dipeptide. And upon submission to the ATB, every molecule is given a unique identifying number, which means you can track it through all the different processes and revisit it many years into the future. So in NMR, it's incredibly important for amino acids to be have their side chains named, their side chain atoms named appropriately. In 1969, a set of rules were recommended by IUPAC for the representation of proteins and peptides. What we developed is a tool that automatically applies these rules to amino acid side chains that have been uploaded to the ATB. As a subset of this tool, if a branch occurs in an amino acid side chain, the two atoms have chain priority assigned to them based on CIP priority rules, and I'll outline what those are now. So every atom is given a number of bonds away from the C alpha atom. So 1 bond away gives you a C beta, 2 bonds away gives a C gamma. But if you have an example that you can see here, there are 2 C deltas. So they are equally bonded away from the C alpha, but they need a chain priority assigned to them. And this occurs in many different stages of this extremely branched amino acid, which is actually derived from the middle residue of vancomycin. So the way in which priority is determined is that the priority of the atom is first. So a heavier atom gets a higher chain priority number. And you can see that here in the branch off carbon theta, where you have an oxygen, which is given chain 1, and a carbon, which is given chain 3. If it's not able to be determined, if both atoms are of the same variety, you then look 2 bonds down and see whether you can find maybe the first bond is the same, but the second bond away, one of them's a carbon, one of them's a nitrogen. And that's a way that it can also be resolved. You just keep working down that train, those 2 chains, until you find a point of difference. If there are no points of difference, however, you can use the atomic coordinates and RS configuration of the branching. That's a particularly, that itself was a particularly difficult part of the code to get right, but we did get there eventually. And in some situations, such as a branch, in the case of rings, an atom can be actually counted twice. You can see here in C lambda 1, it could be considered part of the 4 chain and the first chain, because it's if you count it down, it would be part of both chains. But the first priority is given to the lower number, so it is considered part of the first chain. So this is just a slightly bigger look at how that naming process continues, how that naming process has been applied to this amino acid. And now I'd like to take a little bit of time to talk through the examples that we used throughout this paper and in lab throughout this paper and in the creation of this protocol. The first case we looked at was tyrosine. Tyrosine is 10 residues long and cyclized through a backbone peptide bond. It contains 1 non-standard amino acid in position 9, the ORN here. And what we were able to do was build that as our first proof of concept. Can we build this amino acid in that tripeptide, upload it, get a library file, which we can then append into Cyana, and create an unrestrained, and run Cyana unrestrained to create a chemically feasible structure. And luckily, we were able to do so. So you can see here the different amino acids present, and you can see number 9 sitting quite happily in that chain, something that wouldn't be possible without the implementation of the ATB and Scilib protocol. As I said, it is also backbone cyclized. In order to create that backbone cyclization, we simply used a standard distance restraint file. Anyone who's used Cyana to close a disulfide bond would be familiar with that kind of restraint file. And we simply used that to close, we used a peptide restraint file to close the ring. And that is actually available if you follow through our paper and go into our GitHub. That file is available for download to be used to create backbone cyclizations. We then moved into cyclosporine as an intermediate case. Cyclosporine has 11 residues, 3 of which are non-standard. It also contains 7 backbone N-methylations. Again, it is cyclized through a backbone peptide bond. So the issues with cyclosporine is those 3 non-standard amino acids. And as I said as well, Cyana was not currently able to build any backbone modifications on standard amino acids. So what we were able to create were library files as an additional proof of concept, where we built a standard amino acid and then added a methyl group onto the nitrogen. We were successful in creating that, and cyclosporine ended up again with a chemically feasible structure when we ran it through Cyana unrestrained. Another key part of amino acids that contain non-standard amino acids, peptides that contain non-standard amino acids, is that they often have some form of link between 2 side chains. While Cyana is able to, as a default, create disulfide bonds, there wasn't a process to create a link between 2 other side chains to sort of cyclize a peptide in that method. So we created the Cyana library linker, which with an enormous amount of help from the molecular dynamics group at the University of Queensland. So what we've done is created a process through which you build both sides of the chain, and you build both sides of a linking bond, and then upload it to the library linker. So this is what our library linker looks like. You provide the residue index, so what site of the chain the amino acids sit at, the atom bond, the names of the 2 atoms that form the bond, and overlap pairs. So I'll talk you through this with the idea of a disulfide bridge. We wanted to prove that we were able to build and create a disulfide bridge using our protocol, which is different to the Cyana one, but still form a nice bridge. So you can see if we were to form a link between residue X and Y, a bond would occur between the gamma atoms on both sides of the chain. What you actually upload, however, is built past that extension. So you would upload a residue that captures both sides of the bonds and at least one atom further along the chain from the bond. You would then provide the linking bond name. So you would say for residue X, the bond exists between S gamma and S gamma prime, and between residue Y, again, S gamma and S gamma prime. You would then provide overlap atoms. You would tell the library linker that C beta of residue X is equivalent to C beta prime of residue Y, and likewise with the sulfides and the additional carbon. What library linker does is it then runs through and cuts the excess files off, the excess atoms out of each file, and induces then a link between a linking statement and distance restraint, which can then be appended into your cyana project. And when you run through, it will create a linking bond. So our white whale of sorts with vancomycin, we approach this as an extremely complex case. Despite only being 7 residues long, it has 3 side chain to side chain bonds between residues 2 and 4, 4 and 6, and 5 and 7. So of its 7 residues, 5 were non-standard amino acids. It also had a methylated N-terminal. So we had to produce an N-methyl group, N-terminal, as well as each of those side chain bonds. We were able to successfully create an unrestrained version of vancomycin using our protocol, one that we're extremely proud of because it took quite a while to get right. So that's an overview of all the different aspects of our pipeline. We were very fortunate to be able to provide be provided with the opportunity to practically apply our protocol to a peptide that was undergoing research in the Stromgaard lab. So they are looking at cyclic APP and its potential drug use in dementia pathways. So to perform the calculations in cyana, we used the usual format where the sequence file, chemical shift file, peak lists, and angle restraints were all imported to cyana. We also used our library linker and our ATB library file generator. And what we were able to do was generate a structure for them that had a link between the 7th and 11th amino acids because they were investigating specifically different forms of creating a cyclic side chain. It's a really interesting paper. We were very fortunate to be able to get the opportunity to work with them. And I would encourage you all, if you are interested in this work at all, to have a look at what they've done and how they've been able to use our protocol. So in conclusion, we have successfully integrated Scilab into the ATB. It allows us to produce cyana-appropriate library files, as well as library files for other popular structural determination software. I didn't touch on that in this talk, but if you are a user of CNS, Explore, or ARIA, they all have files that can be mimicked in a similar, which we detail in our paper. We created the library linker, which creates side chain to side chain bonds in order to cyclize these peptides that are extremely biologically active. We've created, to our knowledge, the first automatic script, which will go through an amino acid side chain and assign the appropriate names for the atoms. It seemed we did hope it was something that existed already, given the rules have been around for a very long time. But when it wasn't found, we ended up creating it ourselves, and we are yet to find it, to find a way to break it. So if you do end up following this protocol and find a way that that it seems to be broken, we'd love to hear from you so we can update it. We'd like to address further limitations going forward, such as we can't use this protocol to create beta amino acids. It does have to be your standard nitrogen, carbon alpha, carbon amino acid. So that's something we'd like to address going forward. I'd just like to acknowledge those who worked on this paper with me, my supervisor, Mehdi, as well as Martin and Alan at the ATB, Anne, who was vital for this paper, Peter Gunther for allowing us to use cyana and Scilab and create and integrate it into the ATB, the Christians who provided us with those practical applications, or those practical ones that we could use as a guinea pig for our protocol, and the bioNMR group, which is my group up at CAI here at University of Queensland.